Hello everyone, welcome to Midha Physiology blog, and I am Dr. Sonam. Today we are going to discuss about the juxtaglomerular apparatus. The juxtaglomerular apparatus can be defined as the combination of specialized tubular and vascular cells at the vascular pole of the glomerulus. So what does this mean? When you look at this glomerulus in this renal corpuscle, you can see that from this end, as the tubule is arising, this is the tubular pole, and this side is the vascular pole, right? Now, at this vascular pole, the afferent arteriole, when it is invaginating into the Bowman's capsule, the tunica media layer of the afferent arteriole, the smooth muscle present over there, it is modified. Okay, and this specialized modified smooth muscle cell in the afferent arteriole, you call it juxtaglomerular cell. And just in this region only, we already came to know that when the loop of Henle was ascending up, forming the thicker ascending loop of Henle at its distal end, the columnar cells was modified into, the cubital cells were modified into the columnar cells, which we call the macula densa cell. And we know that this macula densa cells, this region is lying just between the afferent arteriole and the efferent arteriole. So here you see in this region, right, this region which is just near to the glomerulus, so you call it juxtaglomerulus. So this region near to the glomerulus, you see the vascular cells, the vascular smooth muscle cell in the afferent arteriole, it is modified. The tubular cells in the ending of the thick loop of Henle, it is also modified. And in between them there is the mesangial cell. Okay, so these three structures together we are calling it juxtaglomerular apparatus. And you see the component are the modified smooth muscle cell in the afferent arteriole, we are calling it the juxtaglomerular cell. The modified or the specialized tubular cell in this region, we are calling it the macula densa cell, and in between there is the mesangial cell. So you can see the juxtaglomerular apparatus again here. As you can see, this is the afferent arteriole and this is the modified smooth muscle present over the tunica media layer of the afferent arteriole, and this modified cell can even be seen in the efferent arteriole, but it is lesser in number. Okay, so this modified smooth muscle cell, you call it juxtaglomerular cell. The tubular portion, the end of the loop of Henle, there is the tubular portion, the cells are also modified columnar in nature, and these are the macula densa cell, and in between there is the mesangial cell, right? So again, this is a juxtaglomerular apparatus which is near to this glomerulus or the network of the capillary. Okay, now this group of cell, the JG cell or the juxtaglomerular cell and the mesangial cell and the macula densa cell, okay, this group of cells, these are acting together to regulate the filtration of the blood through this capillary. Okay, from the afferent arteriole, when the blood is going and come into the capillaries, from these capillaries the blood was ultrafiltrated. We had already discussed this thing. Now the thing is, as the capillaries are those part of the vascular system where it does not contain the smooth muscle, so the blood flow in the capillary can be controlled, and eventually the filtration of the blood through this capillary into the Bowman's space can be controlled via the constriction or dilatation of this arteriole, either that is the afferent arteriole or the efferent arteriole. Okay, so this constriction or the dilatation of the afferent arteriole or the efferent arteriole, so as to regulate the filtration of the blood in the glomerulus, it can be done via various mechanisms, and one of the mechanism which is present in the kidney itself is the group of this specialized cell, the JG cell, the macula densa cell and the mesangial cell. Okay, so you see this group of cells, functionally they are acting together to regulate the glomerular filtration rate, or the rate at which the blood is being filtrated from this glomerular capillaries into the Bowman's space. Okay, so as you can see, the type of cells present in this apparatus, they are the juxtaglomerular cell, the macula densa cell, and the mesangial cell or the Lassus cell. So even though we are saying it as juxtaglomerular apparatus, it is not a separate apparatus present inside the kidney, but just the combination of this specialized cell. They are working together so that the glomerular filtration through the capillaries can be regulated, okay, in a normal way. Now let us see about this juxtaglomerular cell. You already come to know that it is the modified vascular smooth muscle cell in the afferent arteriole before entering the glomerulus. Okay, now let us see in what way this smooth muscle cell in the afferent arteriole had become modified. It has been found that this smooth muscle cell, when they are modified, okay, they have well developed Golgi apparatus, mitochondria and ribosomes. Okay, so these cell organelle, let us tally their function. When we are talking about ribosome, we know that the ribosomes, these are the protein manufacturing plant. Okay, and this Golgi apparatus, they are the wrapping and the packing department of this cell, right? So obviously when there is a lot of well developed ribosomes inside the JG cell, that means the protein synthesis inside this JG cell is increased, and that is being wrapping and packing more by this well-developed Golgi apparatus, right? And for this synthesis of the protein, the energy is required, and this energy is provided by the well-developed mitochondria present inside the JG cell. Okay, so obviously the JG cell, they are now having much more amount of this protein synthesis, and they are being stored in the secretory granules in the cytoplasm. So these proteins are actually renin. Okay, now this renin has various functions. We know that the renin is eventually helping in the formation of angiotensin 2, right? And this angiotensin 2 is acting in various mechanisms in our body so as to regulate the blood pressure in the long term. Now while talking about the kidney, and in this video, let us look at this function only. Okay, let us concentrate over here only. The angiotensin 2, which is being formed by the secretion of the renin. Okay, this angiotensin 2, they have more receptor in the efferent arteriole. So you see that in the physiological condition, when this angiotensin 2 is being released, they are mostly acting in the efferent arteriole. Now if the angiotensin 2, they are acting in the efferent arteriole, they are going to constrict the efferent arteriole, due to which the pressure of the blood will now go into the glomerulus only. So the glomerular capillary pressure would be increased, and the rate of filtration would also be increased. Okay, so you see the JG cells, okay, which are loaded with the renin. So when the JG cells, they are stimulated, they are going to cause the release of the renin. The renin is eventually leading to the formation of angiotensin 2, and this angiotensin 2 via the constriction of the efferent arteriole will lead to the increased hydrostatic pressure in the glomerular capillaries, such that the glomerular filtration rate would be increased. Okay, now let us see at what condition these JG cells would be stimulated, or what are the function of this JG cell. It is found that the JG cells, they are acting as the baroreceptor and the volume receptor. Okay, now this term baroreceptor, when we are talking about baroreceptor, we know that these are the group of those specialized cells which is becoming stimulated whenever they are given, they are sensing the fluctuation in the pressure. Okay, now the pressure over here is the pressure of the blood. So these baroreceptors are also stress receptor. We already know this thing, and the baroreceptor we have mostly studied in the cardiovascular system. Here in the cardiovascular system, we came to know that whenever the pressure of the blood in the carotid sinus or the arch of aorta, when the pressure of the blood was increased, the baroreceptor, as it is a stress receptor, right? Okay, so when the pressure of the blood it is increased, it is causing the stretching of the blood vessel, right? And this stretching lead to the activation of this baroreceptor, and this activation of the baroreceptor is giving signal to the higher center, so that there will be decrease in the sympathetic activity or increase in the parasympathetic activity. In both way, what they are going to do is they are going to decrease the blood pressure towards the normal range. Okay, so here the baroreceptor was basically detecting the high blood pressure inside the vessel. Okay, so as to cause the response was the decrease in the blood pressure towards the normal range. Now when you are talking that the JG cells, they are also acting as the baroreceptor, remember very clearly that the JG cells, when they are acting as baroreceptor, they are not going to detect the high blood pressure inside the vessel, but they are going to detect the low blood pressure. Okay, so whenever the pressure of the blood in the afferent arteriole is decreased, at that time the JG cells, they are stimulated. So whenever there is decreased renal perfusion pressure, at that time the JG cells, which is functioning as baroreceptor, they would be stimulated, and this stimulation would directly lead to the release of the renin, which is present in the cytoplasm of the JG cell, and this renin would now cause the formation of the angiotensin II. This angiotensin II is a very potent vasoconstriction. We know that thing. And this constriction in the efferent arteriole is eventually leading to the increase in the filtration of the blood. Okay, so you see, this baroreceptor, now we can classify that the baroreceptor can be of two types, right? The high pressure baroreceptor and the low pressure baroreceptor. In the case of the cardiovascular system, this baroreceptor, they were detecting the high blood pressure. So they were the high pressure baroreceptor. But the JG cells, they were detecting the low blood pressure inside the afferent arteriole. Okay. So when there is a low blood pressure inside the afferent arteriole, eventually the pressure of the blood reaching towards the capillaries, the glomerular capillaries, are also decreased, and the rate of the filtration of the blood is also decreased. Okay, so here when these JG cells are sensing this all thing, they are releasing the renin, and this renin is helping in the production of angiotensin II. This angiotensin II, in the physiological way, in the physiological criteria, okay, this angiotensin II, they are acting over the efferent arteriole so as to increase the glomerular filtration rate. Okay, so you see here how the JG cells, they are acting as a baroreceptor to maintain the glomerular filtration rate. Okay. Now it is also found said that the JG cells, they are also acting as volume receptor. Okay, so how? Because whenever the blood volume in the afferent arteriole, it is decreased, or whenever the hypovolemia is there, then again these JG cells acting as a volume receptor would detect it, and they again are going to cause the release of the renin. Okay, and this renin via the angiotensin II is now going to regulate the glomerular filtration rate. So you can see this hypovolemia and the decrease in the blood pressure in the afferent arteriole, they are related with each other, because whenever the blood volume is less, the pressure would also be lesser. Okay, so more or less these stimuli are similar, which is going to cause the increase in the renin secretion so as to form the angiotensin II and to regulate the glomerular filtration rate. Okay, now the other thing is the hypovolemia, or the decrease in the blood pressure. At that condition in our body, we also know that the activation of the sympathetic nervous system would occur. Okay, now interestingly these JG cells, they are also innervated by the sympathetic nerve. Okay, so whenever the sympathetic nervous system is activated, they are going to release the norepinephrine, and this norepinephrine, they will act via the beta-1 receptor, which is present in the JG cell. Okay, and the action of the beta-1 receptor on the JG cell, they are again going to cause the secretion of the renin, and this renin via the production of the angiotensin II again helps to regulate the glomerular filtration rate. Okay, so overall you see the juxtaglomerular cells acting as baroreceptor or acting as a volume receptor, they were maintaining the glomerular filtration rate. Okay, so the next component in the juxtaglomerular apparatus is the macula densa cell, and we had already discussed that these are the specialized renal tubular epithelial cells located where the thick ascending limb of loop of Henle is continued as distal convoluted tubule, and this portion is lying between the afferent and the efferent arteriole. Here the cells, they are converted into the columnar in nature. Okay, now the macula densa cell, they are found that they are acting as the chemoreceptor. Okay, so these macula densa cell, they are stimulated by the sodium chloride load present in the ultrafiltrate. So let us see this thing over here. So again you can see this is the afferent arteriole, right, through which the blood is going into the capillaries, and from the capillaries the filtration would occur. And this is the modified smooth muscle in the afferent arteriole, which we call the juxtaglomerular cell. And in this region only the cells in the ascending limb of loop of Henle, the thicker portion, they are modified as columnar cells. You see, these are the columnar cells in this tubular portion, and these columnar cells, they are called the macula densa. Okay, now when this blood is filtrating, coming from the afferent arteriole and then filtrating from the capillaries, they are going into the Bowman's space and into the proximal convoluted tubule, loop of Henle, and the ultrafiltrate is now finally coming into this macula densa region. Okay, now this ultrafiltrate can have the sodium chloride in the increased amount or in the decreased amount, and this macula densa cell would now get stimulated by this tubular salt or the concentration of the sodium chloride present in the ultrafiltrate, and eventually they would now give signal towards the JG cells or towards the afferent or the efferent arteriole, okay, so as to maintain the glomerular filtration rate in such a way that the sodium chloride filtration coming or the sodium chloride load coming up to the macula densa cell would be in normal range again. Okay, so let us see this thing in detail now. You see, this is the macula densa cell. Okay, the macula densa cell. So this is the lumen portion in which the ultrafiltrate, it would come from the descending limb of loop of Henle into the ascending limb and then into the macula densa. So this is the luminal portion. And then you see, we also know that these three structures, they are together. Okay, the macula densa, this is the mesangial cell, and this is the smooth muscle of the afferent arteriole or the JG cell. These three structures are lying together, and this three structure we had called it the juxtaglomerular apparatus. Okay, now when the amount of the sodium chloride, okay, coming in this ultrafiltrate, it is higher, then this macula densa cell, what it is going to do is the higher amount of the sodium chloride would now get reabsorbed into the macula densa. Okay, so how? Because of the presence of the sodium 2 chloride potassium co-transporter present in the apical or the luminal side of this macula densa cell. So when lots of sodium chloride is coming, they would be reabsorbed by the co-transporter, that is sodium 2 chloride and potassium co-transporter, and then this sodium and the potassium and sodium is now being reabsorbed back into the circulation with the help of this sodium potassium pump, which is present in the basolateral side. Okay, now you see, when lot of sodium chloride and the potassium, they are being reabsorbed, the rate at which it is being disposed out into the circulation may not tally. Okay, so lots of sodium chloride may be now accumulated inside this macula densa cell only. So when lot of sodium chloride is accumulated in the macula densa cell only, via osmosis, again the water will also start to get reabsorbed into this cell. Okay, now there is water, there is sodium chloride. Okay, all together the macula densa cell will start to now swell. Okay, and then the another feature which is present in this macula densa cell is in its basolateral side there are the channels. Okay, this freely moving, this arrow portion is basically in this region there is the presence of channel. And when we are talking about channel, we know that the channels, they are associated with simple diffusion. The channels can be ligand gated or voltage gated or mechano sensitive channels, right? So here the ligands present over this basolateral side, they are mechanosensitive channels through which the ATP or the adenosine, they are being diffused outside into the interstitium. Okay, so what does this mean is, whenever the sodium and chloride were reabsorbed excessively due to more amount of sodium chloride coming in the ultrafiltrate, now there will be more functioning of the sodium potassium pump in the basolateral side also, so that the sodium chloride will be reabsorbed. Okay, now due to more functioning of the sodium potassium pump, the ATP may be hydrolyzed into the adenosine. Okay, now this adenosine would now get out of the cell into the interstitium via this channel. This channel is the mechanosensitive channel. So we also know that the rate of this disposal of the sodium is not equivalent to the rate of its reabsorption from the ultrafiltrate, due to which the macula densa cell was swelling. So when this swelling would occur, the stretching of this cell will occur, and then this mechanosensitive channels, which is for the adenosine, okay, so these channels would get open, and then the adenosine would now be released into the interstitium. This adenosine, they would have the receptor in the smooth muscle present in the afferent arteriole. Okay. Now, when this adenosine, they act to its receptor, that is A1 receptor. This A1 receptor is a G protein coupled receptor with two kind of subunit. If they are acting via the GQ subunit, we all know that now they are going to cause the activation of phospholipase C, and then there will be the secondary process inside the cell in such a way that there will be production of the inositol triphosphate and diacylglycerol, and with the help of this inositol triphosphate inside the smooth muscle, the calcium would now increase. Okay, now this increase in the calcium inside this smooth muscle is going to cause the contraction of this smooth muscle or the contraction of the afferent arteriole as a whole. Okay, now in this way, whenever now the vasoconstriction would occur, okay, whenever the afferent arteriole will undergo vasoconstriction, then obviously the blood flow towards the capillary would also decrease. When the blood flow towards the capillary would decrease, then the amount of the blood filtrated through the capillary would also decrease. When the amount of the blood filtrated through the capillary is decreased, the amount of the sodium chloride present in the ultrafiltrate would also decrease. Okay, so eventually here you see the stimulus was increase in the sodium chloride load, and the response was vasoconstriction, and finally it will lead to the decrease in the sodium chloride load in the ultrafiltrate, or the sodium chloride load would not be excessive coming up to this macula densa area. Okay, so here you see that this increase in the sodium chloride load is eventually going to cause the afferent arterial constriction, and it is again going to regulate the glomerular filtration rate. Okay, now this was one of the function in which whenever the sodium chloride was loaded in the ultrafiltrate via this mechanism, via excessive production of the adenosine and its action over the AT1 receptor present in the smooth muscle, it was causing vasoconstriction. Okay, now this AT1 receptor, I told you that it can have the GQ subunit and also it can have the GI subunit. Okay, via GI subunit it is causing the inhibition of the adenylyl cyclase and decrease in the cyclic AMP, and eventually the renin, which is present inside this juxtaglomerular cell, it would not be released. Okay, so when the renin in the juxtaglomerular cell would not be released, the formation of the angiotensin II would be lesser. That means in the efferent arteriole its function would be lesser, and ultimately it is also helping in the regulation of the glomerular filtration rate. So here I would like to tell you that how the renin and its subsequent product, this angiotensin II, is helping in the regulation of the glomerular filtration rate. I will subsequently discuss in next video. So this thing I have written again in this slide. The increased sodium ultrafiltrate when delivered to the macula densa cell, there will be increased adenosine formation. This is now going into the interstitium, and they are acting on the AT1 receptor present in the smooth muscle cell so as to cause the vasoconstriction via the increase in the calcium in the smooth muscle via this GQ subunit. Okay, as the AT1 receptor is a G protein coupled receptor, and via the vasoconstriction the GFR would be decreased, and whenever the glomerular filtration rate would be decreased, now the amount of the sodium filtered would also be decreased, and the amount of the sodium chloride coming up to the macula densa area would also be decreased. Similarly, via the decrease in the renin release also, it can regulate the glomerular filtration rate. So again you can see here the stimulus was increasing glomerular filtration rate. When the glomerular filtration rate was increased, the amount of the sodium chloride being filtrated is also increased. So there will be increased sodium chloride concentration in the tubular fluid in the loop of Henle, and this increased sodium chloride concentration, when it is reaching up to this macula densa area, there will be the formation of the adenosine. This adenosine would act over the afferent arteriole smooth muscle cell. Okay, because this area are in close proximity, they would act via AT1 receptor so as to constrict this afferent arteriole, and whenever the constriction of the afferent arteriole will occur, now the blood flow in the capillary would be decreased and the GFR would be decreased or come back to normal range. Okay, so in this way you can see one of the stimulus for the active for the functioning of this macula densa cell was increase in the sodium chloride load. Now let us see what will happen if the sodium chloride in the ultrafiltrate is decreased. Okay, so here supposedly if the sodium chloride which is coming into this macula densa cell, it is decreased, then what would happen? So if the sodium chloride load coming in this ultrafiltrate is decreased, this whole functioning, okay, the sodium chloride potassium, this co-transport functioning would be decreased, the production of the adenosine itself would be decreased, and instead the macula densa cell would now stimulate some another mechanism. Okay, inside the cell there is a vast cellular pathway which would be various kind of cellular pathway which would be stimulated inside the macula densa cell. The eventual result will be it is now going to cause the production of prostaglandin E2. Okay, now this prostaglandin E2 would now go and act over its receptor, which is again present in the smooth muscle, this modified smooth muscle of the afferent arteriole. When the action of the prostaglandin would occur through its receptor, okay, in this smooth muscle or in this JG cell, okay, then the renin which was present over here, it would be released. Okay, when the renin would be released, then there will be increase in the production of the angiotensin 2. This angiotensin 2 would now act over the efferent arteriole. With the construction of the efferent arteriole, now the glomerular filtration rate would be increased, and when the glomerular filtration rate would be increased, the sodium chloride load coming in the ultrafiltrate now would be increased in such a way that this stimuli was previously the decrease in the sodium chloride load, and now because of the increase in the glomerular filtration rate, that stimuli would be compensated. Okay, so this was one mechanism. The other thing is the decrease in the sodium chloride load in the ultrafiltrate. It is also causing it is also helping the macula densa cell. Okay, in this condition, the macula densa cell also gives signal so as to cause the activation of the nitric oxide synthase for the production of the nitric oxide. Okay, now this nitric oxide is going into the afferent arteriole, and you know that in the afferent arteriole, since it is a vasodilator, it is going to cause the dilatation of the afferent arteriole. So whenever the afferent arteriole dilatation would occur, then obviously more amount of blood will flow into the capillary. So when the more amount of blood will flow into the glomerular capillary, the filtration rate would eventually be increased. Okay, so you see, whenever the decrease in the sodium chloride load was occurring in the ultrafiltrate, let us recall what may cause the decrease in the sodium chloride load. If the glomerular filtration rate is decreased, then obviously the amount of the sodium chloride in the ultrafiltrate would also be decreased. Okay, so this is one of the reason. Apart from that, whenever there is a more reabsorption of the sodium chloride from the proximal convoluted tubule, this can also decrease the sodium chloride load in the ultrafiltrate. But let us here focus in the glomerular filtration. Okay, so when there is the decrease in the glomerular filtration rate, there will be decrease in the filtration of the sodium chloride also, and there will be decrease in the sodium chloride load coming in the ultrafiltrate. And when it is being sent by the macula densa, it is now causing the afferent arterial dilatation, or via the renin, it is now causing the constriction of the efferent arteriole so as to maintain the GFR. And whenever the GFR is increased or maintained, then the amount of sodium chloride which is being filtrated would be coming back again towards the normal range. Okay, so you see, the renal tubule, okay, from the macula densa cell, the signals, they are going into the glomerulus. The macula densa cell, they are sensing either decrease in the sodium chloride load or increase in the sodium chloride load, because the macula densa cell, they are functioning as chemoreceptor, and whenever they are signaling this thing, they are going to stimulate the filtration rate from the occurring from the capillary. Okay, so in case if there is the increase in the sodium chloride load via the adenosine, it is going to cause the afferent arteriolar constriction, or whenever there is decrease in the sodium chloride load via the production of the nitric oxide, the same afferent arteriole will also dilate. Okay, now in this mechanism also there will be alteration in the renin secretion so as to maintain again the filtration rate. Okay, so overall you can see that the sodium chloride, the tubular salt, okay, is going to stimulate the macula densa cell. So this macula densa cell is a part of tubule. So according to this stimuli coming into the tubule, the tubular portion is giving the feedback towards the glomerulus for the maintenance of the glomerular filtration rate. Okay, so this feedback mechanism arising from the renal tubule, that is from the macula densa cell, going to the glomerulus for the proper filtration of the blood to occur, that is the tubuloglomerular feedback mechanism. Okay, so this was about the macula densa cell, and this third component was the mesangial cell. This mesangial cell, we already know that it is a parasite-like cell, and then it is present between the afferent and efferent arteriole. And this mesangial cell, they are contractile in nature, so whenever this mesangial cell they are contracting, the glomerular filtration rate would also decrease. Okay, and this mesangial cell also have renin, so whenever this renin secretion occur, then obviously it is going to cause increase in the production of angiotensin 2, and this angiotensin 2 would again now regulate the glomerular filtration rate. Okay, now I have given the detail about this mesangial cell in my third video. The topic of that third video was the blood filtration in the nephron. Okay, you can go through the third video so as to know in detail about the mesangial cell. So yes, we have discussed. So what have we learned today? Today we have learned about the juxtaglomerular apparatus. Okay, now why we have given separate terminology to this group of cells present nearby the glomerulus? Because this group of cells, this specialized modified group of cells, whether that is from the tubular side or whether that is the specialized modified vascular smooth muscle cell, this group of cells, when they are acting together, okay, they are going to cause they are going to regulate the filtration of the blood through the glomerular capillaries, because the capillaries itself, they cannot regulate. That capillary filtration can be regulated either via the afferent arterial constriction or dilatation or via the efferent arterial constriction or dilatation, and that thing in the kidney level is being done by this specialized group of cell. Okay, that are the JG cell, the macula densa cell, and the mesangial cell, and these cells have been termed as the juxtaglomerular apparatus. Okay, and how the macula densa cell is acting in various kind of stimuli, whether that is increase in sodium chloride or decrease in sodium chloride load, and how the JG cells, they are operating as the baroreceptor, the low pressure baroreceptor, we have also covered in this video. Hope it was useful to you. And the main thing, okay, so main thing which is released from this juxtaglomerular apparatus, main protein was renin. So how this renin is helping in the formation of angiotensin 2, and the role of angiotensin 2 in the regulation of the glomerular filtration rate. Okay, that thing I'll be covering in the next video. So if this video was useful to you, please guys like, share and subscribe to my channel. And yes, this much for today. Thank you.